and find Escherichia coli, and that is organism 20. So everything will be labeled organism 20 in the back. Now, when you're doing labeling, I would suggest you write E. coli just so that you don't have to later on go then and figure out what number 20 was. Obviously not a huge thing today because we're only working with one organism, but in the future we will be working with more than one organism, so you'll want to make sure you're grabbing the right numbers in order to do the experiments. The next thing that I would like to draw your attention to is on page 9. Whoops, there it went. That is your inoculating labels, right? So there are 3 pages of inoculating labels. Each of these will be used, so you cut it out and you will use a rubber band to attach it to all tube media. Petri dishes, because they're disposable, you can write right on the Petri dish. But anything that is in a tube, so that would be a slant, a deep, or a broth, you're going to use a inoculation label. And there are plenty there for you to use for the entire semester. So that was what I was saying. You could bring scissors if you want, keep them in your personal box, right? Because you're going to have to cut out inoculating labels daily to use them. And then if you would roll forward to, right, so table of contents, aseptic technique, and microbial ubiquity. That's how you figure out where we are today. And then I do want to give you a little bit of introduction as to the common areas of the lab, right? So first of all, instructor bench. So this will be where I do things, where I demo stuff for you. Back bench is that bench back there. That is where you will find all of the supplies that you need to do your experiments, but you do not perform experiments on the back bench. You go there, you pick up your supplies, you bring them back to your desk. So typically those supplies will be shared either among your whole table or among your team of 4. So for instance today, each team of 4 will get a plate with E. coli, a broth with E. coli, and a slant with E. coli. So if you have decided that it's going to be you, then you 2 do not go get E. coli, because if you do, then there will be a team that doesn't have any. So designate one person from your team of 4 to go back and get those supplies and bring them back. You will each go back and get all of the other things that you need individually, right? But the organisms are shared. Student storage, right? You're already kind of getting used to that. That's your personal box. Personal box will remain there. Please don't put your name on it. Just use the box that corresponds to your number. And that's really helpful for us, because if you accidentally put a communal supply in your personal box, like let's say you open up your stuff and you're like, oh, my striker's gone. And so I look and I go, okay, you're number 17, Tuesday, Thursday, AM 17, I'm going to check their box and see if they accidentally put the striker there. So that's why we do it by where you sit. You don't go into other people's personal boxes. You will let me know if something's missing, and I will go and look for it. So that's where you keep lab coat, all of your supplies, right? You can bring your colored pencils, you can bring scissors, your Sharpie goes there, all that fun stuff. And then you have either a community drawer or a community drawer / box. So table 3 and table 2, you guys have just a drawer. This is our ADA compliant. You might have noticed it's a little shorter, so that someone, if they were in a wheelchair, could access this table. But that means your drawer is really short and it won't fit everything. So you guys, in addition to what's in your drawer, have these boxes. So only students 1 through 8 need to worry about this, but that's where lots of your communal supplies are. So that would be your stains, your Bunsen burner, which is going to be used today, your striker, which is going to be used today, and they go back into those areas. Nothing that belongs to you belongs in that drawer. If your personal belongings, like your inoculating loops, go in that drawer, it's going to be in the garbage by the time you get back. So make sure that you don't put anything that belongs to you in the communal drawer. Your things go in the personal box, hence the name, personal box, community drawer. All right. If you put things in the wrong place, make that a mess, like you spill stain all over the inside of it, and you just go home and you don't say anything, and then tomorrow morning someone says, someone spilled stain all over the inside of my drawer, then you might lose some of those technique and cleanliness points that we talked about on Tuesday. So this is our lab space. This is our incubator room, right? Incubator room, fun acoustics. You cannot hear anything out here. We can hear everything you say in there. So just keep you're talking smack about your teacher, don't do it in the incubator room. Right? In the incubator room, we have goggles. We have a shared group of goggles. So you don't have to have your own goggles. You can have your own goggles. If you choose to use ours, they are sterilized when you get done using them, and I always sterilize them when I get here, right? Just in case the other instructor forgets, I turn it on and sterilize it before I come in. But in this room, our incubators, ours are labeled Wagner, because that's me. So 37 degrees, which is body temperature. Below that is a 25-degree incubator. That's room temperature. So don't flip out if it's off, because room temperature is room temperature, right? So body temperature, and then we have a fridge over here. We have one shelf. Ours happens to be the bottom shelf, and that's our 4-degree incubator. That's where we store things. We grow things at body temperature or room temperature. We store things at 4 degrees. Inside of each of those are test tube racks that are labeled table 1, table 2, and table 3. You guys will put your stuff in the one labeled table 3, table 2, and table 1. In addition to that, there are Petri dish racks that are labeled with racks, table 1, table 2, table 3. Now imagine, right, there are 24 of you. Each of you today will have 5 broths and 1 plate. That's over 100 tubes. So you can imagine when you come in on Tuesday, if you know you only have to look in one rack, that makes it easier to find your stuff, right? So put your stuff in the appropriate racks, and that just makes it easier for you to find it later on rather than you have to sort through everything, right? So make sure you put them in the right place, and whenever we're done with things, right, we discard them in the morgue. We don't take them home as souvenirs, right? Because even though they're biosafety level 1 and 2, we still don't mess around with them, right? It's very important that you always label culture so that we know what's in tubes, right? And it's important that you either completely fill out an inoculating label, or you put that same information on a Petri dish, right? So you don't just put your name and like some random thing that nobody understands. You put your name, you put the date, you put the organism that's in there, and the media that it's in. So as a rule of thumb, right, when you're writing on a Petri dish, just think about these 4 things and make sure those 4 pieces of information get there. Now you can put your initials, that's totally fine, but you need to put your name so that we know that it belongs to you. Anything that is not labeled will get thrown away, and you will come in the next day looking for your stuff, and it won't be there because I will throw it away, because it is unsafe for us to keep anything that we don't know what it is, right? So make sure you do that. And one thing that I'm going to demo for you, right, but you don't want to tilt your tubes. When you tilt them, you can spill what's in them. If what you're spilling is bacteria, that's not good, right? All right, so today we are not going to be using any of our chemical stains, so we're going to breeze through that. We'll talk about that Tuesday. We are going to talk about some waste handling because as a practice, we're going to start using the morgue today. So when you are done, and everyone in your team is done, and you have E. coli to discard, we're not going to discard the plates, but we are going to discard the tubes. So we will remove the stickers. So when something goes into the morgue, there should be no labeling on it at all. That just saves our lab tech some time. She's got 7 sections that she has to clean up after. So you'll take the stickers off, you'll walk over to the morgue, and everything is really labeled, so if you're ever like, oh, where does that go, right? We have small test tubes. What you're working with today are all small test tubes. So you'll take the labels off, put them right here, and then this will get autoclaved. So Emily will come in after class, she will wheel this into the room, and she will sterilize those things before they go into the trash, right? So we don't send live organisms into the trash. So what I will do over the next couple days is as we're using areas in the morgue, I will show you where to put things. But for today, the only thing that's going in the morgue are these test tubes of E. coli that you've been taking organism out of and putting over here. Right? So that's the morgue, we call that the morgue, right? The rest of that stuff we'll talk about later because we don't want to overwhelm ourselves with information when we're not even going to be using that information today, right? So, I think that's probably all that's in here. Yes, yes, yes. Oh, I should say, right, non-hazardous, so paper towels that you use to clean the countertops, paper towels that you use to wipe your hands go in the trash. They don't go in the morgue, right? Because those are not contaminated materials. All right, so that is that. That's all of our little lab prep stuff. So let's talk about our labs that we're going to be doing today. You have 2 labs to do today. That is not uncommon. It is very common that you will have to prep and perform more than 1 lab a day because a lot of these labs take very little time to finish, and we want to make sure that you're exposed to as many techniques as possible. We will not always perform them in order, right? Because I've done this lots and lots of times, I have found some ways of saving time, right? And one of those ways that we can save time today is by starting the day's lab with lab exercise 3, because some of you have to let something sit out for 30 minutes. And you know what is a sad microbiology student? One who does everything and then at the end of lab realizes I now have to do a 30-minute something, where I sit around and wait for 30 minutes. So we will do this first, just in case you are one of the people that has to do that 30-minute thing, right? So lab exercise 3 is called microbial ubiquity, right? We know we hate big words. Ubiquity is not a science word. It's a regular old word. It means everywhere. So we're going to test that theory. You are going to be exposing petri dishes and broths to all sorts of different environments to see if there are microorganisms present. Because microorganisms are microscopic, we can't see them unless we grow them. So we are going to grow those on petri dishes, and we are going to grow those into broth, right? So if you look at your lab manuals, in lab exercise 3, there is a table on page 36 at the very top, and there are student numbers. If you don't know your student number, look at the drawer right next to you and then see that number. Look on that table for what that corresponding number says to expose your plate to. So, expose it to the air for 30 minutes. That just means you open it, leave it sitting on the counter, and 30 minutes later you close it, right? Bathroom sample. That means you get to go to the bathroom and you can either like brush the counter off onto it, you can take a cotton swab and swab some area of the bathroom and then rub the cotton swab on the surface of the agar, right? Whatever you want to do. Fingertips touching the media. So you take your dirty, filthy hand that you think is clean and you touch the surface of the agar with it. See if anything grows. Right? Blow dust on it. So you can blow dust or you can like find a little dusty spot. Right? Moist lips pressed against the agar. We'll see how clean your lips are. And you just go, give it a little kiss. Right? Coins or cell phone, right? So you can just take some inanimate object if you don't want to take, if you don't have a coin or you don't want to, touch your phone. You can just take something and touch the agar with it. Hair combed over media. Obviously, like I couldn't do that today, right? Because I wore my hair in what my son calls them panda buns, right? So maybe you can swap with somebody or ask somebody, can you just like shake your hair over my, right? See what falls in it. And then we have vigorous cough onto blood agar. So only a few of you are going to be using blood agar. Guess how you can tell the difference between TSA and blood agar? It's red. We have to bleed sheeps to make this. So don't use it if you have not been told to use blood agar. I mean, we don't personally bleed sheep, but that's how we get blood agar, right? Which is a really important medium because it allows us to tell whether bacteria are able to eat red blood cells, which is an important pathological consideration. So if you have been chosen, and I think it's 3 of you, you will cough vigorously. So don't be a baby about it, right? You're gonna like open it and go and try to cough what is on your pharynx out onto the plate. So give it some vigorous coughs. And then all of those plates are going to go agar side up, which feels upside down, but it's not. Agar side up into the incubator. The reason they go agar side up is when I did this, stuff might have fallen on here. And if I put it like this, stuff can fall onto the agar and contaminate it. So if I do this, nothing can fall into the agar. So we always inoculate agar side up. Now, when I do this, right, I label it with just a pen. So I look at there and I go, what do I say? And then the date, and then I put the organism, which is just my cough, who knows what it is, right? And then media would be blood agar. And notice I write just along the outside rim of the agar, right? Because I don't want to write all over the bottom and then I can't see what's growing there. And then I take it inside and I incubate it and I come back and I get horrified because things grow in my throat. But hey, guess what? Microbes are ubiquitous. They are everywhere, right? The second thing you do in this lab is do a broth swab, right? So I think it's evens. So the evens touch your body and odds touch some spots. Cool. So here's what you're going to do. You're going to grab a cotton swab, right? If you're odd, not, you personally, you're odd number, you'll use one of these because we don't care that they're not like super, super sterile. If you're going to touch your body, you can use one of these that are sterile. What ultimately you are going to do is actually leave that swab in here. You can tell it's too big right now, right? So what you're going to do is break it so that this is shorter than the tube. And then if you're touching some part of your body, you can actually take it, right, and get some of that broth, and you're like, ew, gross, but it's sterile, right? And then you're like, let's just see what my neck can grow, right? So you rub it all over your neck, and you just drop it right in there, and you close it. And then we will see what grows on your neck. And we're actually going to use these tubes to gram stain next week. We're talking about gram staining, right? So we can differentiate between gram negative and gram positive. Back to my dog, is going to love it when I get home. I can smell the chicken broth. So that's lab 3, right? So it's pretty quick, right? Like that just took me like 5 minutes or 10 minutes to describe. It's going to take you less than that to do it, right? Then you're going to move to lab 3 or lab 2. Lab 2, you are going to be performing these 4 aseptic transfers, right? So that means moving E. coli and only E. coli from media to sterile media, right? And you're going to do a slant to slant transfer, a plate to broth transfer, a broth to slant transfer, and a broth to broth transfer. So you will need 3 inoculating labels, and we're going to do something unique here that we don't usually do. Because we have 4 different kinds of E. coli, we want to differentiate on this label which of those E. coli it is. Right? So for your broth, the organism would be like my neck. For these, if I'm labeling this slant, I'm going to say my name, the date, and I'm going to say E. coli slant, because that's where it came from. And then for media, I can just put TSA. I don't have to put slant because guess what it's sitting on? The slant, right? That's not unknown to you. What is unknown to you when you pull it out of the incubator is, which of these slants is it? So to differentiate, you have to say this one is the E. coli slant, this one is the E. coli broth. So just today, because we have more than one E. coli, you will say E. coli and what kind of E. coli it is. Does that make sense? So normally you would just put E. coli, because it doesn't matter. But because today you're doing slants, plates, and broth, I want to know where they came from. Now, why is that? Because if you end up with a contaminated E. coli, I would like to know, did you contaminate it, or was it contaminated when you got it? And the way that I can tell is if multiple people in your team, everybody's contaminated that came from here, then that probably wasn't you, it was probably already contaminated. If you don't tell me which one it is, then it makes it harder for me to track it down. So today and today only, you are going to, for this one, say E. coli slant, E. coli plate, E. coli broth, E. coli broth. Oh, I said something backwards earlier, right? So broth to slant, broth to broth. So when I say organism, E. coli slant, E. coli plate, E. coli broth, E. coli broth. When you do those transfers, right? To work aseptically does not mean to work sterilely, which always freaks everyone out, right? But, I mean, you know that when you have surgery, it's not sterile either, right? You think, right? It's not, are you sterile when you have surgery? No. Even when they rub you down with betadine, you're not sterile, right? Nothing is sterile. The people that walk into the room are not sterile. What they are doing is aseptic surgery using sterile tools, and that is exactly what you're going to be doing. The first step in that parade is cleaning off your countertop, right? So we're going to spray the countertop, we wipe the countertop down, and then we work in an area with a Bunsen burner, right? So we light our Bunsen burner. If it doesn't spark, let me know because we can actually change that little flint that's inside there, right? I want to work near the flame because flame makes heat and heat rises. And all of the animalcules will hit the hot air and go like this and go away. So we work here near the flame of the Bunsen burner We also use the Bunsen burner to sterilize the tool. So I am not sterile, I'm clean. I've washed my hands, the area is not sterile, it's clean. This needs to be sterile. Just like if you're having surgery, the scalpel is sterile. The glove that holds the scalpel is sterile because that gloved hand might actually touch the inside of your body, right? So in order to sterilize, you need to get this end of the loop red hot. So you want to make sure that your flame is blue because blue is hotter, right? Notice it's getting red hot. And now it's sterile. The longer it sits outside that flame, the more likely it is to be contaminated. So we want aseptic transfers to be as quick as is safe. So if I'm doing the first one, slant to slant, I will grab my E. coli slant. This is a one-person job. It's awkward. You need to get used to it. You're going to take this, which I've called your dominant hand, right, which for statistically most Americans is your right hand. You're going to take your pinky and hold that, and you're just going to hold that in your hand the whole time. Notice it's always, now you're not going to talk over it, right, that's going to contaminate it. But if this is facing down, nothing can fall in it, right? If I set it on the countertop, something can fall in it. So I sterilize, I open, and as soon as I open my tube, which I have not done because I'm talking to you, I wave this through the flame. I'm not trying to sterilize this, I'm trying to create hot air again. When I wave that through, it creates a movement of air out of the tube so that stuff from the air can't fall in it. And then I take my, quickly sterilize, I know this is taking much longer, right? But pretend I just sterilized it. I wave it through and I touch the surface, right? So I'm just gonna go touch, just like that. And then I wave it through and I close it. And that's when I let go of that. Now it's on the end of my loop. I grab what I'm going to, which in this case is a slant. I grab it with my pinky, I wave it through, right, creating that outward movement of air, and I take that loop and I just go over, 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 over. So I'm not pushing into the auger, I'm just moving it over the whole surface. My goal here is to get that entire slant covered in bacteria. Now I wave it through and close it because I want this to be open as little as possible. And my last job is to sterilize this before I put it down. So I put it back here. And if you have organism on the end of the loop, it will actually make a little flame sometimes because it will ignite like a piece of wood, right? So you do that, sterile, boom, done. So if I'm working from broth, similar circumstance, right? I grab my broth, I have my sterile loop, I grab it with my pinky, I wave it through. When I do this with a broth, what I like to do is kind of stir up the bottom because bacteria settle. And then you want a loop full in your loop, right? It's kind of like when you blow bubbles. You can't blow bubbles if there's no soap across your bubble wand, right? So you want a bubble full, and if you're going into broth or into slant, it would do the same thing, rub it all over the surface. If I'm going into a broth, I do this, and I just give it a little tappy, tappy, tap, and wave it back through, close it, and sterilize it. Yeah?
After you sterilize it, how long do you leave to know that it's cool?
It cools pretty quickly. One thing that you can do, especially like whether you're going into a slant or a broth or a plate, you can take, right, so this is my organism and this is my hot sterile loop. I can do this and leave it for a one potato. And now I know it's cool, right? So when it went in, it was hot, but the cool liquid will cool it. So you can do that with him. You can also do that with the slant. You can actually just touch the surface of the slant, wait a second, and then grab some organism. But they cool, the metal that's used in these loops heats very quickly and it cools very quickly. So it will cool pretty quick, and they're actually a lot more tolerant of heat than we are. So even if you touch it and you're like, woo, that's still really hot, it doesn't necessarily mean it's too hot for them. Now, last thing that you're going to be coming from is dish, right? We always work with Petri dishes upside down for the same reason that I just said, right? So stuff that's on the lid doesn't contaminate them. So I am going to choose a colony from this area out here where I can see individual little colonies, right? Not over here where they're all crowded, but over here where they're by themselves. And I go, okay, there's one, right? So I sterilize my loop, make it red hot, right? And as to your concern, I can go like this. Just find an area and go and stick it right in the auger, and I hear it sizzle. And then I just touch the top of the loop, and I lay that back down. And now I'm done. I didn't flame this, right? Because it's plastic. We don't flame plastic. Plastic will melt, right? Also, notice I'm not wearing gloves. Gloves will melt. So now I grab whatever I'm going into, right, which in this case it's broth to slant. So I wave it through, and I go over the whole surface, and then I wave it through, and you're done. You sterilize this. You shut off your Bunsen burner. You take off your goggles, because now all of the organisms are closed. And you take this test tube rack, you walk to the incubator, you put your 4 test tubes into table 1 rack, table 2 test rack, table 3 test tube rack, and you're done. Then you come clean up, right, put everything back, put the E. coli over there, put the stuff back over there, take all of your stuff, put it in your personal box. And the last thing, right, after the lab coat is off, you spray the counter down, you wash your hands, and you exit the room. So when you're finished, you're free to go. You don't have to wait for everybody else to get done. If you get done before your lab mates, please do not discard the organisms, right? The person who is last on your team, just give them the organism. Don't assume they're done. If you're done, just leave them in someone else's test tube rack. So that's your goal today, right? So your goal today is a plate from some environmental sample and a broth from either an environmental sample or a body sample into 37 degrees. And these 4 inoculations, 2 slants, 2 broths labeled with inoculating labels into the 37 degree rack. And right when we're taking and putting, you might have noticed that I went over and I threw away all the big rubber bands, because I think big rubber bands are a really bad idea when you're doing this kind of stuff, because big rubber bands you have to wrap around multiple times, right? Try to keep this upright, which is awkward, right? So you're going to wrap a piece of paper around it, and then you have to go like this and hold it upright and make your hands really awkward. But if you tilt it, you're going to tilt organism all over your hands, and then you have to start all over again, and we have to decontaminate you, which is not a huge deal, but you have to wash, and we have to do stuff. So tubes upright, right? Why am I saying this? You think it's because I don't know any better, but I'm telling you, after doing this for 40 semesters, right, statistically, people always turn it sideways and dump the bacteria onto themselves. So there are some little tiny test tubes back there, or rubber bands. There are not many, right? They run out quick. But make sure everything is labeled, and when you're clean, you're good to go. And any questions? Yes?
What's the difference between a slanted agar and a plate agar?
So slant is this, and this is this.
Right, no, I know.
Yeah.
I'm just wondering
So
why
you so this I would use when I want to isolate an individual organism. This I would use when I want to grow a bunch of organisms. So we'll get to use them in their